So hello, everyone. Welcome to the next installment in our Illumina Genomic Discoveries Educator Speaker Series as a part of DNA Day's month of events. Illumina and Biotech California Institute appreciate your partnership and participation in our programming. We hope that you find today's event and all webinars in the IGD Educator Speaker Series helpful in furthering your own learning and find it useful for growing your collection of curriculum and education resources. We hope that you feel empowered to take what you learned today back into your classrooms as we work to make life science, biotechnology, and genomic curriculum more accessible for all educators and students. In the chat, you will find links to soon find links to both the Biotech California Institute website and the Illumina website that contains information about a multitude of resources that are available to you and your students. If you have any questions about these resources, please reach out to myself or the Illumina team. This session is being recorded and will be available on the Biotech California Institute YouTube channel, along with the rest of the Speaker Series webinar recordings after this event. We are honored to have two keynote speakers with us here today. At the end of the presentations, you will have the opportunity to ask both of our keynote speakers questions. Please feel free to interact with the speakers as they give their presentations. Most likely they'll be making use of the chat. And please feel free to post any questions that you have for them in the chat as well. We do ask that you save questions until the Q&A session at the end, but please feel free to post them in the chat at any time, and I will ask them during the Q&A session for you. So with that, I would like to introduce our first speaker, Dr. Priyam Singh. Dr. Singh is a bioinformatics senior scientist at Illumina Inc., where she develops state-of-the-art cloud-based applications and computer programs to analyze large quantities of genomic data to answer biological hypotheses. Dr. Singh received her PhD from Boston University, where she used genomic data to stratify subtypes of lymphocytic leukemia that are hard to distinguish under the microscope and have different treatment types. Dr. Singh is also a 200-hour certified sky breath meditation instructor, certified by the nonprofit Art of Living Foundation. In her free time, she volunteers to teach conscious breathing techniques to help still the monkey mind. She is also the leading investigator in running a randomized controlled trial looking at epigenetic changes in normal human subjects due to sky breath meditation intervention. She is using Illumina's technology of whole genome bisulfite sequencing in this study. Dr. Singh, I will spotlight you here and I will pass it over to you.
Thank you so much, Michaela. How is everybody doing? I don't see too many faces. Are you all feeling a bit shy? Do you want to turn on your cameras? There we go. Bharti just turned on her camera. Yeah, this is good. Now we can interact and have fun. You know, I want to hear from you. What is your biggest challenge with mental health before I begin my presentation? And you can put it in the chat or you can share. You know, what are you facing? You know, a big problem with your own mental health. Does anyone want to share? We can make this interactive. You know, it'll be so boring if I just keep talking. I have a presentation here which can bore you to death if we don't interact.
This is, oh, sorry.
Yes, go ahead.
This is Juanita Nunez. I'm a STEM teacher at the elementary school in San Isidro. And one of I see a lot of mental health with the staff, and that energy is being projected to the students. As teachers, we are overwhelmed, and I don't know if teachers are aware of the strategies that we can provide them, like mindful breathing. I know that you're a guru on the breathing, which I'm trying to bring that into all classes. As a STEM teacher, I start with mindfulness and then I go into my STEM activity.
Oh, wow.
So one thing I do notice is the teachers are being burned out and they're transferring. They're so frustrated that they're projecting that energy to the kids. Their kids are bringing their own issues with whole COVID struggles in their community. So to me, I'm interested in how can I bridge that gap between teachers and students.
Wow, that is wonderful. Thank you so much for sharing that. Yeah, I think you just hit it exactly. You know, we ourselves have anxiety. We have, in the chat, you can see this stress and anxiety. Pandemic has not helped, right? It's made everything more difficult for us. And then you all are, you're the role models for our children, and they have their own stress, and then they're bringing it here. So what I'm going to start with is a very small, short meditation using breath work, and that's going to ground us, and then we can begin our presentation. So if you can, sit with your spine erect and just sit comfortably. Doesn't have to be in a specific position. If you're sitting on the chair, just be comfortable. Have your spine erect. Just feel relaxed and keep your eyes closed. If you don't feel comfortable keeping your eyes closed, you can just dim your eyelids so you're not really looking at it. Take a deep breath in. Hold the breath and let go. One more time. Fill your lungs with as much breath as you can. And let go. You can put your right hand on the belly and just become aware that when you breathe in, the belly rises, expands. And when you breathe out, the belly contracts. And it just happens naturally on its own. So one more time with the hand on the belly. Take a deep, long breath in. Hold the breath. Hold it as much as you can. And let go. So your attention is on the belly as it's rising and falling. Once more, breathe in. Fill your lungs, fill as much air as you can. And let go. Do this 2 more times on your own. Slow the breath down as much as possible. Like you're taking a sip of wine very slowly. Become aware of your feet. Ankles. Become aware of your thighs. Hips. Your stomach. Your chest. Your neck. Become aware of your hands and shoulders. Become aware of your face. The head region. Become aware of your whole body. Your whole body. Feel that stillness and silence. Emanating from every cell of the body. Take one more deep breath in. And let go. One last time. Fill your lungs with the breath. And breathe out. And slowly and gradually, you can open your eyes. How did that feel? You can put in the chat. We did a very, very short mini meditation. Was that good? Yeah. Okay. So now that we are grounded and we are relaxed, I'm going to begin the presentation. So you can see my slides? Yes? Okay. So, you know, what I want to tell you about today is Illumina's technology and how we do sequencing of the microbiome and how the microbiome is actually so related to mental health, the latest research that we have. So as a bioinformatics student, as a bioinformatics scientist, what I do is we have tons and tons of genomic data that comes out of our machines. And when I say tons, it's billions and billions of nucleotides. It's, you know, humongous in terabases. The problem now is not that we can't sequence, we are sequencing too much. And so people like me who are, you know about computer science and algorithm development, and they need to come back and analyze the data so that we can make sense of the data. Yeah. And there's different ways that you can actually sequence the microbiome. And Molly here is going to be talking about a paper actually, where they do 16S ribosomal RNA gene profiling. And she will actually go into more details there. But really there, what you're doing is you're taking your bacteria, archaea, and there's the ribosomal gene, the 16S RNA, which you then sequence and you cluster. And eventually you find these OTUs, which are, you can think of them as signatures of the specific genus of the bacteria. And what this tells you, what this helps you in is it identifies the OTUs. So it really helps you identify what are the species of the bacteria, which is really the most important thing, right? But besides this, you can do other things too. There's something called shotgun approach, where basically you take all the microbes and you take their DNA, and then you do something called de novo assembly, where you basically cut up the DNA into small parts and then you put it all together. And remember, the microbes also express genes, so you can also do metatranscriptomics, which is looking at what are the genes that are being expressed by these microbes. So it's a very versatile technology. And you know, essentially, we can now begin to take a human being or a mouse or a rodent's microbiome and then use the 16S profiling or shotgun approaches to identify the specific species of bacteria that are there. So actually, wait, I want to go through. Okay, I cut that slide out. You know, just really quick, I think this will be a fun thing for you to tell your students when you talk about them. That what I'm going to say is not on the slide, but there's a lot of fun facts about microbiome. There's just so many of them. There are 10 trillion microbes in your body. And 95% of these microbes are in the gut. And the size of this is, you know, almost the weight of your microbiome in your gut is equal to the size of your brain. And, you know, they're all over our body, all on the skin, they're in the openings, nose, mouth, they're in, you know, mostly in the stomach, but also you can find them in many other organs of the body. And really, you how many cells we have as an adult, 10 times that number of cells are in the microbiome. So it's like a whole community, living inside your body that you don't even know of. They're just invisible. And they do so much for us. They help digest our food. And now we're finding out that there are many possible pathways of how the microbiome talks to your brain. And here there are 4 ways that I'm showing here. The first one is that, these microbes interact with immune cells in the gut, which then prompts the cells to make cytokines. And these cytokines, they circulate from the blood to the brain. So essentially interacting with the immune cells. The second way is through the microbes interact with gut cells, which are specifically called endocrine cells. And these cells produce neuroactive molecules and peptides, which then interact with the vagus nerve, which sends signal to the brain. So the vagus nerve is the biggest nerve in our body. It runs all the way from the brain to every part of the body. It has its tangles on, and then it's really big in the stomach region. And one of the ways we think the second brain in your stomach interacts with your main brain up here is through the vagus nerve. And how that happens is through these neuroactive chemicals. The third one is metabolites. The third one is just metabolites like butyrate, and these are produced with the help of the gut microbiome. And these also, they can cross over the blood-brain barrier and then alter the cell activity at the barrier itself. And then finally, there's been one report of a study from University of Alabama where they actually found bacteria in the brain. It's a controversial study, and it's, this is a very active area of research right now. So, I want to tell you about this one very, very special study. There's a lot of studies with mouse and rat that have been done where we know that these bacteria are playing a very important role in autism, depression, anxiety, Alzheimer's, bipolar disorder, and whatnot. But this one study, the one that I'm showing you here, Lactobacillus reuteri, it is actually shown here where it is stuck to the gut. And this is a very interesting study because the scientists weren't really looking for this bacteria. What happened is they fed mice a really high-fat diet, and so these mice became really, really fat. And their kids, they ended up being autistic because these mice were so fat, obese, right? And so these kids weren't interacting at all. They were showing very high symptoms of autism. And they put these kids in a litter with normal mice and their kids. And mice usually they eat each other's poops. And so what they found is that these kids, these mice babies that were exhibiting autism symptoms, they reversed completely, and they started bonding socially, and the disease just disappeared. And so then they were like, what happened? We didn't do anything. All we did is we put these mice together. What happened? And they started probing more and more and more. And then they found out that there's this one specific bacteria called Lactobacillus reuteri, which is responsible for promoting social bonding. So if you go on the internet, and Molly will tell you how to go on the internet and find these kinds of papers, you will find that with rat and mice, there's a lot of studies that implicate the microbiome in mental health. But when it comes to human subjects, there's this one project called the Flemish Gut Flora Project, where they took 1,000 human samples. And what the authors found is that there are 2 specific species of bacteria called Coprococcus and Dialister, which are consistently depleted in individuals that have depression. So there's a very high correlation that if you're depressed, you don't have these 2 bacteria. And there's a lot of research in this area is happening. There are companies being formed, and there's also a lot of hype. So how to solve human disease problems using bacteria, right? So that brings me to, why I'm so fascinated by breath, because, I have always been really interested in increasing productivity and focus, and, doing my work in as little time as possible. And what I found out is that meditation is the answer, or breath work is the answer. But it's very hard to meditate, right? When you tell your mind, come on, stop thinking, or do something, or don't think negative, or don't get anxious, it's really hard. But when you bring in breath work or breathing like we did just now, we see that it's much easier for the mind to settle down. And why is that? This is because the breathing activates the parasympathetic nervous system. And that is the system that brings a sense of calm to the system, because it's opposite from the fight and flight. Yeah, it automatically. So if there's one thing you want to remember from here, well, there really should be two things. So the first thing should be, whenever you feel anxious, or you feel like your emotions are taking over, take deep, long breaths, and invest in learning a breathing technique or meditation technique that really clicks with you. Because breathing can help lower heart rate, it can help reduce stress, reduce inflammation, increase memory and recognition. And we just experienced how we barely did 5 minutes of breathing, and how it completely switches the way the brain is. And the breathing, the deep breathing, it also activates the vagus nerve. And the vagus nerve is also connected to the parasympathetic nervous system. So in many ways, the microbiome is connected to the vagus nerve, and breathing slowly also activates your vagus nerve. So all of that is connected to each other. So I want to quickly tell you, I know I'm running out of time. I want Molly to have enough time. I want to tell you about the study that I'm so excited about. So what we're going to do is, using Illumina's technology, this is the first human study where we'll be looking at how sky breath meditation, which is a form of breathing meditation technique, how it changes the epigenome. So to tell you a little bit about the epigenome, your DNA is what you have inherited from your parents, and the DNA never changes unless, of course, you're having mutations and things like that. But then on top of the DNA, there are these chemicals that are deposited. And what happens usually is that the chemicals will prevent the DNA from opening up. So if you have DNA and you have a very important gene, let's say my ring here is a tumor suppressor. If you're healthy, this gene is going to be expressed. But if you've been really stressed out, having really bad diet, too much sugar, not sleeping enough, there might be a chemical that will come and suppress this gene. It won't let this gene open up. And this is epigenome. Epi means on top. And so when you do breathing and meditation, or you change your exercise, or you improve your diet, or you suddenly start to sleep, what happens? The first thing that happens in your genome is your epigenome begins to change. So we're going to be looking at how the epigenome is changing with sky breath meditation in 50 total subjects, 8 weeks of intervention. So after you learn sky breath meditation, you get a home practice that takes only about 15-20 minutes to do. So these people will do it for 8 weeks. We'll take their blood, we'll do the sequencing, and we think that genes that are responsible for inflammation, cancer, immunity, aging, all of these are going to show up as being changed. And another very cool thing is we're going to compare these profiles of methylation patterns with those from other diseases. So we also think that there's going to be a protective effect from this meditation in preventing other diseases. So we're going to be doing all of that. The study is being written up, and I am just so super psyched about it. So this is my last page. That's last weekend. We were at a festival where we were putting colors on each other. So that's my family. I welcome you to check out artofliving.org. It's a nonprofit, and you can learn sky breath meditation there. It's only something like 8 hours of your time to learn it, and then you get a home practice. You can also email me or text me if you want to know more about anything I have said today. I would absolutely love to interact with you on this topic. And with that, I will let Michaela take over.
Thank you so much, Priyam. This was fantastic, and I know I've been writing down questions, so I'll have plenty for you here shortly. But with that, I'm going to jump us back to this slide here briefly, as I'd like to introduce our second speaker. So Dr. Maddie is a postdoctoral researcher at the Salk Institute, where she studies how microbes impact animal behavior. Dr. Maddie received her PhD from Duke University, where she used zebrafish to study new treatments for the infectious disease tuberculosis. Originally from rural Northwest Florida, Dr. Maddie is a passionate educator, science communicator, and mentor. In her free time, she enjoys going on long walks, traveling, and watching Star Wars. Dr. Maddie, I will pass it to you here, and I'll spotlight you in just a second.
Awesome. Thank you so much. All right, so I hope everyone has their critical thinking caps on. I know it's late in the day, you've been working really hard. So I'm gonna hopefully share with you some techniques to really engage with scientific literature like that Dr. Singh highlighted, and the type of work that I'm doing. So instead of really telling you about my work, which I'm gonna call maybe giving you a fish, I'm hoping to teach you how to fish or how to engage with this literature. So I am Molly, and a postdoctoral research fellow at the Salk Institute. So I'm going to briefly tell you my trajectory into science, what's brought me here. Then we're going to really dive into finding science articles that are reputable and figuring out how we really know what we know. And I'm going to start a quick timer just because I am so long-winded that I just always have to keep an eye on myself. We're going to take a little dip into reading microbiome papers and engage with what the research is showing us. And then I'm going to briefly tell you how I do microbiome research and how many of us can also do it. That allows us to go beyond just associations. So I received my bachelor's degree from North Carolina State University, and then got my PhD in genetics and genomics from Duke University. I'm now at the Salk Institute as a postdoctoral research fellow, and I'm happy to announce that in fall of 2022, I'll be starting as an assistant professor in the Department of Biology at the University of Portland in Portland, Oregon. So if you have any students who are looking to get out of California and go to a school in Oregon, send them my way. One thing that I always like to remind you and your students is that PhDs in STEM fields are free and you get a stipend. So this means you don't have to go into debt to get that higher education you might be seeking. And a postdoc was something I had no idea what it was when I was in undergrad, let alone high school. So a postdoc is really a great training position between a PhD and the next step. Maybe that next step is a professor position, maybe it's industry, maybe it's something in science communication, outreach, patent law, whatever. It's a really great way to get more training. So I'm really interested in how microbes impact host behavior. And as Michaela said in my introduction, my background is actually in pathogenic microbes. And one of my favorite pathogenic microbes is this one called Toxoplasma gondii. And Toxoplasma infects rats or other rodents and actually changes their brain chemistry and brain structure in a way that makes them less afraid of cats. This is really cool and will actually allow the parasite to get into its preferred vector, its preferred place to live, which is inside felines. So not just pathogens can change your brain chemistry, even commensals or non-pathogenic microbes that exist in and on us, as Dr. Singh shared, there's a lot of them. They've been associated with making mice more social, and other signatures of microbiome composition in human infants are associated with fear behaviors. And these two examples, as well as hundreds of other bodies of literature, support a microbiota gut-brain axis. And maybe you've engaged with some of these ideas and maybe you've done some Googling. So if maybe you've done a little bit of Googling or you're interested in this field, could you maybe type into the chat if it's safe for you to type, what is the most complicated or frustrating part about trying to find or engage with scientific literature? So just take a moment to just think what's stopping you from reading my science or Dr. Singh's PhD work or anything like that? Feel free to type it into the chat. Yeah, science jargon is definitely intimidating. There's a few other issues that go beyond the scope of what I can talk about today, meaning maybe paywalls or inaccessibility. But I know that when I go onto Google and I type in things like microbes, mood, mental health, I usually get this first little bit that says scholarly articles. And that's usually where I go. But I know that if my mom maybe Googled gut health, mental health on her Google settings, she might get a whole bunch of ads about companies that are being started, or maybe, finally a few that have some connections to universities. And finally, maybe you've heard that probiotics are good for your gut. Maybe you're going to Google probiotics for mental health, and now you're getting a bunch of ads and things to buy. And this can be overwhelming when all you really wanted to do was understand how to make your gut healthier, or how do we know what makes our gut healthier? So I'm going to tell you how to find reputable science resources. Yes, yeah. So there's some more things in the chat that say it's difficult to find the full literature, and now Google is a little less specific. Absolutely. So I love starting with popular science. I engage with a lot of pop science books, news articles, and radio shows. So I would suggest starting from reputable resources. This might be university press releases, NPR, magazines like Scientific American, or professional societies like the American Society for Microbiologists. Then you can type in something into Google, like maybe we're here in San Diego, UCSD Microbiome Mental Health News. I did that, and I found this article from last year that said, "Wisdom, Loneliness, and Your Intestinal Multitude," which kind of sounds clickbaity, but we read a little further and we actually see that this is based off of research going on at UC San Diego. I encourage you to engage with that article, figure out kind of what you think it's going to be about, but then within it, find the link to the original work, which you can do usually in the very first sentence of a well-written article. So here we find the original article in Frontiers in Psychiatry, and it's called Association of Loneliness and Wisdom with Gut Microbial Diversity and Composition, an exploratory study. And with that article, we're going to take a very shallow dive into how to read this, find main points, and think about what these data show us. So the very first part, don't worry, you don't have to read this whole slide. We're actually going to share a link to this paper at the end of the presentation. But the very first place you start when reading a scientific article is the abstract. And even if this article is behind a paywall, you can read the abstract and get a lot of information. The thing I usually care most about is why this matters. That's usually going to be the first sentence and the last sentence. So loneliness is a health concern. Then you're going to find the goal of the work. So here it was to investigate the association of loneliness and wisdom with the gut microbiome. Then we'll get into how did they do that? Well, so they did some surveys on 184 community dwelling individuals. They did some self-reporting, and then they took fecal samples, yes, poo, and profiled them using 16S rRNA sequencing, which I'll describe in just a moment. And then what they found was some associations of the richness or diversity of the microbiota in terms of these two terms called alpha diversity and beta diversity, which I'll explain. And I'm hoping that with these few key terms, we can give you the tools to engage with a lot of scientific literature about the microbiome. But before we get into that, you're probably thinking, what is wisdom and loneliness in a scientific sense? I also had that question. So I went into the methods section of this paper and found that they administered 7 different surveys to all of their participants. And these surveys have been established by social scientists, psychiatrists, and other mental health experts to give metrics of things like loneliness, compassion, emotional support. The authors of this paper did not invent these methods. They were pre-existing. And this is great because most microbiologists, myself included, are not professionals in developing surveys to understand depression. And so if you're not going to use pre-existing surveys and data sets, it's really important for us as scientists and members of the world to collaborate with interdisciplinary colleagues to kind of fill the gaps in our knowledge. Okay, now to get into things that I actually know a little bit more about. So this is the main tool that we use as microbiologists to really understand what is the composition of a microbiome. The way we do this really easily, because we're not going to dive deep down into your gut, we're just going to collect some poo. So this poo contains a bunch of microbes. They might be living, they might be dead, and those microbes all have DNA inside them. There's a specific part of DNA that Dr. Singh mentioned called 16S. You can think of this just as a name tag. And this name tag lets us basically say, in this poo sample, what proportion of the name tags are, say, gaga. So we have this poo, and we see 60% of these name tags are gaga. And we're actually getting percentages rather than whole numbers. Like I can't tell you there's 3. I would just be able to tell that there's 60%. Whereas this poo sample would only have 25% of the whole microbial community being gaga. And what we do with that is we create these really beautiful bar charts that help us see within this one individual what proportion of their microbiome are these different species of bacteria. So we could compare individual A with individual F, and we can see that A and F look pretty different, right? And now we can go B, C, D, E, and we think, okay, cool, there's some variability within person A and B and C and D. And then where the data starts to get really informative is when we take these individual microbiome compositions and we start to sort them into the groups based on those surveys. So maybe these 3 individuals scored really highly on that wisdom test. These are the wise individuals. And these individuals over here maybe scored really high on the loneliness survey. So maybe these people, microbiomes might be associated with loneliness. But how can we prove that? What sorts of things are we going to actually look at instead of pretty little bars? So the 2 main ways that we think about microbiota composition, just to begin engaging with what it looks like and how they're similar or different, are alpha diversity and beta diversity. So alpha diversity, you can just remember, is the richness within a sample. So A, alpha, one sample, or sample set. So here, let's call these 3 individuals the wise ones. And we see that they're pretty evenly distributed. These 8 species are fairly equal in amounts across these individuals. Whereas this lonely group is not as evenly distributed, where this purple species represents a larger proportion of their microbiota. So what we can say here is that this wise group, or an individual in this wise group, has higher alpha diversity because they're evenly distributed and there's a lot more of them. Beta diversity lets us look at differences between samples, B between beta, easy to remember. And so this lets us ask, how similar or dissimilar are these samples? So again, if we're just clustering by these arbitrary designations I've made of wise and lonely, we can see that they have 4 shared species, right? They have the purple, the pink, the purplish pink, the purple, the orange, and the green in common. But then this wise group has the gray, the beige, the yellow, and the green. So they're unique species. And what we can do using complex statistics that even I don't fully understand is really compare the metrics that we've collected from surveys and see if any of those answers can actually explain the microbial differences. So say this person had high emotional support, are they going to be more or less different in their microbiomes than people who had low emotional support? So let's just engage with the 2 main figures in this paper. One is trying to answer the question of, does alpha diversity correlate with loneliness? If you're a high school teacher, you think correlations, you're probably going to think scatter plot or XY chart, right? That's exactly what they do. So they plot alpha diversity. And so the higher the alpha diversity, it means the more species are there and maybe the more evenly distributed they are. And then this loneliness score based on those surveys I explained. And so what this means is, say this individual here, if you hopefully can see my cursor, their alpha diversity is about 22 and their loneliness score is about 20. Now let's take this individual here, where their alpha diversity score is around 15 and their loneliness score is over 50. So what these authors of this paper have concluded is that the less diversity in the microbiome, meaning fewer species present or less evenly distributed, the more lonely that person is likely going to score. Now we can also ask the beta diversity, how much of the difference between these groups is explained by the qualities we've measured. And they do this by showing us how much variance or how much difference between the microbiome is explained by the scores on these tests. So there's this total wisdom test that actually explains about 0.04% of the variance in the microbiome. And this wisdom, this cognitive wisdom one, is a little over 3. And so what we're seeing here is that wisdom and compassion and these other metrics explain about 1% of the difference in the microbiota. And you might be looking at these graphs and think to yourself, what does this mean? Is this valuable? And I want you to do that. I want you to critically think about all of the data that you engage with and that your students are engaging with. So think about how we're measuring loneliness or wisdom and if those are good metrics. Remember, this is self-reporting surveys and found in the methods section. Then ask yourself, are these differences meaningful? You found a difference, but like, is 1% of variance important? Maybe, maybe not. So they conclude that higher levels of loneliness and lower levels of compassion, wisdom, social support, and social engagement are associated with decreased diversity of the gut microbiome. But you might be asking yourself, well, I might have heard that decreased diversity of the gut microbiome is associated with other things like inflammatory bowel syndromes, depression, or obesity. So this is where it's really important that scientists like myself and Dr. Singh control our data sets for age, sex, BMI, and mental health issues before we start going into these cool associations we're interested in. But always something to think about, like, did they correct for people that maybe have IBS? And then in the last minute I would love to talk about designing an experiment to test this more directly, or how we would do this in the lab. So I like to use models for microbiome research. I use animal models like zebrafish, worms, and some of my colleagues use mice. There's other ones that are like squid, even a cheese rind. And if you're opposed to using animal models, you can do all of this in a computational setting or using just chemicals in a vat. The main way that you get at is the microbiome important is by looking at germ-free animals. So you just eliminate all the microbes and you say, are these animals behaving like they are depressed, or are they behaving normal when they do or do not have microbes? And Dr. Singh pointed out this vagus nerve, which is a really important connection between the gut and the brain. You can actually, in mice, get rid of the vagus nerve connection and see if you still have that mood altering behavior you're interested in. You can use the microbiomes from human feces and put them back into another system. Or what I do in my lab is use natural microbiomes, where we isolate bacterial species from the wild in wild animals and put them back into the lab. And I do this using a worm. It's a microscopic worm called C. elegans or Senior elegans. And they're a really great model system. I am running out of time, but I'll just tell you that they can make decisions. They eat bacteria, so they're always just filling up their gut with microbes. They digest some of those microbes, they poop them out, they can reproduce and have really cool mating behaviors, and all of their tissues can age and grow old. And so I'm really interested in taking these animals and exposing them to different microbes and seeing how their eating habits change, their decisions change, and even how their reproductive and aging strategies change over time. So lastly, I just want to encourage you and your students to engage with microbiome research in a constant process where you're finding reputable resources from pop science to primary literature. You're engaging with a few key terms. You don't have to know everything. 16S, alpha diversity, beta diversity will take you so far. Critically think about what you're actually reading. Do the data support the claims? Is it meaningful? And then keep going. Investigate this further. Look for mechanistic work. Stay up to date and just complete this cycle over and over again. With that, I would like to thank you all, Biocom and Illumina, for inviting me. And if you have any questions about what I talked about, please feel free to email me here or check out my website. I work with a great group of people at the Salk Institute, receive some amazing funding and support from collaborators and other great affiliative groups. So thank you so much for your time.
Thank you so much, Maddie. That was incredibly informative. And now what I'm going to do is spotlight Priyam and Molly together. There we go. And now we are here for Q&A time. So this is your chance we wanted to provide a little bit more time than we've had in the past to ask questions and cater this session to your guys' specific needs. So we have two wonderful experts here. Please feel free to raise your hand or post your question in the chat. And at this time now, I will stop the recording. So that way you can feel that you have the space and privacy to ask the questions that you would like.